Okay, good evening for everybody. Welcome to ALAGO webinars. So, today we'll have the lecture Lipid Biomarkers in Paleoenvironmental Studies with Dr. Sebastiaan Rampen. But first I'll have some announcements from ALAGO Association, okay? First, we'd like to call everyone to join ALAGO. Please follow the social networks, Facebook, Instagram, LinkedIn, and also can subscribe to the YouTube channel to participate in other webinars. Now I take the advantage to everyone here to invite to the next ALAGO event in June, next June. It will be about analytical methods to geochemical solutions. We also call all of you to be an associate doing the affiliation in our website, ALAGO website, okay? Second, I'd like to thank the president of ALAGO, Professora Débora Azevedo, the scientific committee, and the technical support from Vinicius, Laércio, and Luisa, who make this event possible. We also thank the sponsors. So finally, we have been successful in the last 6 presentations, and now is the turn of biogeochemistry and environmental issues using organic geochemistry tools. I'd like to thank Dr. Sebastiaan Rampen from the University of Gottingen, Germany, for the presentation of this evening. He will describe different approaches to identify, validate, and apply lipids as tools for the reconstruction of a wide range of environmental conditions, including temperature, salinity, nutrient availability, aridity, and anoxia. Before he starts, I ask just a minute to give a brief summary of his curriculum. So after finishing his bachelor degree in analytical chemistry, Dr. Sebastiaan Rampen was hired as technician by the Netherlands Institute for Sea Research, the NIOS, on a 4-year project entitled Chemical Forces of Diatoms for Age Determination of Petroleum, Improved Tools for Solving Exploration and Production Problems. When this project ended, he continued work on a PhD project with long chains diols as paleoproductivity proxies, which stemmed from some of the results from his work as technician. After completing his doctoral research, he worked on various biomarker projects at NIOS and Utrecht University. And since 2018, he is employed by Georg August University of Gottingen, Germany, where he is working now on the project Isolation and Cultivation of Source Organisms of Long-Chain Diols Lipids from Marine Environments to test their application in bioclimate studies. So, Sebastiaan, thank you again to be here with us, and feel free to start your lecture.
Okay, thank you. I would like to thank the organizers for inviting me to give this presentation. I consider it an honor, but it's also a challenge because I was asked to give an overview about lipid biomarkers in paleoenvironmental studies, but there is no way you can cover a topic that's broad in only 50 minutes. So what I'm going to do is I'm going through some of the results of my previous research, and I'm going to mix it with some of the results of other studies. And I hope in this way I can give, well, like asked, an overview of how lipid biomarkers can be used for climate reconstruction. So most of my work is about identifying biomarkers in marine environments, which can be used for climate reconstruction. And the reason to look in marine environments is, well, one of the reasons is because there is a lot of marine on Earth, so there's a lot of environments which can be reconstructed. But also, maybe even a more important reason is on terrestrial environments, you often have to deal with erosion. So the top soils are eroded and flooded away, and it is difficult to get records of climate. And these topsoils, they may be transported via rivers also to the marine environment. So if you pick a nice location close to a river, you might even be able to reconstruct both terrestrial and marine environments at the same time. So I'm going to talk about lipid biomarkers, and one way you can use biomarkers is by the presence of biomarkers. So when you find a specific lipid, it may be an indication of a certain environment. You can look at the abundance of specific biomarkers, so that can be the absolute abundance, or it can be the relative abundance of one biomarker versus another one. And another topic which is very important in paleoenvironmental studies is isotope composition of biomarkers. And I'm going to talk about these three different topics in my presentation. So in my work, I look at different kinds of samples. So one is environmental samples. So where do we find specific biomarkers? In which areas? In which times? Under which conditions, and what do they tell us? And if we find specific molecules which are promising as biomarkers, then we can see if we can isolate and culture the organisms that produce these lipids, and then we can do culturing experiments, so grow the organisms under specific conditions like different temperatures, different salinities, different nutrient contents, and see how the lipid composition changes with these conditions. And if biomarkers are promising, you can look in the sediments, see if you can find them back, and see if indeed they can be used as climate reconstruction proxies or not. So when I started my work, I was hired on a project for lipid biomarkers and diatoms. And diatoms are a group of algae which evolved relatively recently. And at the moment, these are the most abundant algae on Earth. And they evolved in a time period which was considered a useful period for the petroleum industry. So what we did is we looked at cultures from what we hoped all important groups of diatoms to see if they produced specific lipids which could be used as biomarkers. So one of the first findings are highly branched isoprenoids, or also called HBIs. Already previously, it was known that these molecules are produced by diatoms, but with our work, we could show that there are two specific groups of diatoms, one called the naviculales and one is the rhizosalinid diatoms, which produce these lipids, and the rest of the diatoms do not make them. And analyzing over 400 different marine sediments and 81 petroleum samples, we found that these molecules only occur in samples with an age of less than 92 million years ago. And knowing this information, you can use these biomarkers as age diagnostic biomarkers, which means is that if you find these lipids in your sediments or in your oils, you know that organic material has been produced less than 92 million years ago. This is interesting for the petroleum industry, but it's also an important knowledge for people who are interested in algal phylogeny. If you look at the phylogenetic tree, as you can see here on the screen, so most on the left is the time when diatoms originated. So this is the position where the first diatom common ancestor evolved. And when you go to the right, it's like a time scale. And the more you go to the right, the younger the sediments, the the phylogenetic tree, and the tips of these branches, they indicate species at the current time. So it is thought that DNA mutates at a constant rate, and in that case you can use the branch length as an indication of time. You can average the branch length of a phylogenetic tree, and now if we assume that the rhizosalinid diatoms are the ones that are the oldest HBI producing diatoms, we can assume that the point where these species evolved, that this was 90 million years ago. Then we can draw a line and make a time scale. And with this time scale, we can calculate back the origin of other groups within the diatoms, but also on the broader scale. There are several other age diagnostic biomarkers, like several sterols. There are the oleanan, which are biomarkers from flowering plants. And all these biomarkers, they indicate different time scales, which can be used by the petroleum industry. So that's one of the things we can do with highly branched isoprenoids. But there is more. For example, there's a specific molecule, HBI, with one double bond, and it is used as a proxy for ice. The source organisms of this molecule, these are ice diatoms. And in the picture, in this photo, you can see a brown layer, which consists of algae. So these organisms, they live inside the ice. So at times when there is no sea ice, these organisms do not live, and you won't find these lipids in the sediments. At times when there is permanent ice, well, maybe these organisms, they are living in the ice, but since the ice doesn't melt, the organisms are not released, and then again, you won't see these lipids in the sediments produced at that time. And only at times when there is seasonal ice, so when there is ice building in winter and when there is melting in spring and summer, the organisms, they can live in the ice. When the ice melts, the organisms are released. They sink into the sediments, and you will find the biomarkers in the sediments that are formed at that time. So here is a study by Marcelo in 2008. It is from a core from the northern Icelandic shelf, and it is a sediment core which spans the last 1,000 years. And in this figure, you see a red line, which is temperature reconstruction based on diatoms. In the bar at the lower part of this figure, you see a red and blue bar, which indicates in red periods with a relatively mild climate, and in blue it is harsh conditions. And the black line you see in this figure is the relative abundance of the IP25 biomarker. So what you can see is in the medieval warm period, the relative abundance of these IP25 is low. And during the Little Ice Age, you can see higher concentrations of this IP25, which indicates that it might actually be a pretty good marker for seasonal sea ice. So that's one application of biomarkers. Another where you can use these biomarkers is, for example, biomarkers by sulfur bacteria. There are sulfur bacteria are photosynthetic bacteria which live under anoxic sulfidic conditions. Because they are photosynthetic, they need sunlight, so they cannot live that deep in the waters. And the purple sulfur bacteria, they produce a molecule, it's called Alkanone. So when you find Alkanone, you know that there were sulfidic anoxic conditions. And these purple sulfur bacteria, they live at the depth of less than 25 meters and probably less than 12 meters. And then there are other organisms, other sulfur bacteria, like the green pigmented green sulfur bacteria, which may live slightly deeper, up to 20 meters, and they produce chlorobactene. And then there are the brown pigmented green self bacteria, which also live under euxinic conditions, and they can live up to 100 meters depth, and they produce isoriniurethane. So if you find either of these molecules in your oils or in your sediments, you know that there were periods of euxinia at the time these sediments or oils were formed. And the type of molecules may tell you something about the depth of the anoxic zone. These lipids can be found in samples of more than 1 billion years old. So yeah, I think that's pretty cool. Back to the diatoms. Another group of diatom species called Proboscia, they produce these kinds of lipids with straight alkyne chains with a methylated alcohol group at the C1 position. A methylated fatty acid group at the C1 position, or an alcohol group at the C1 position, and an alcohol group in the mid-chain position. Proboscia is a diatom which lives, for example, in the Arabian Sea, and there it is suggested to be a species related to upwelling. In the Arabian Sea, there are strong monsoon periods. In the summer, when heating of the Tibetan plateau, it causes a low pressure area, and then you get strong southwest monsoon winds over the Arabian Sea. And these winds, they create something which is called upwelling. During this period, nutrient-rich, cold, deep sea waters, they come to the surface, and because they bring that much of nutrients, they cause a strong primary productivity in this area. In the winter, the snow cools down the Tibetan plateau. It causes a high pressure area in this above Asia, and you get winds in the opposite direction. So you get the northeast monsoon, and these winds, they cause a strong mixing in the area. So we wanted to test if long-chain diols can be used as proxies for upwelling, and we looked at 5 different locations where we had sediment traps. So in this picture here, it's a picture of a sediment trap. So the yellow part is like a large funnel, and under it you have like a carousel with sample jars, and one of the jars will be positioned under the funnel, and every after a period like a couple of days or even weeks, the next sample jar will be put in the position. So you can collect samples over weeks or even months. And these sediment traps, they are put into the sea on a long line. At the bottom of the line, you put a large weight, which keeps it attached to the sea floor. Then there's a float on top of the line to keep the line horizontal. And then you can put the sediment traps at specific depths. And material will slowly flow down, get into the funnel, into the sample jar. And after all sample jars are filled and sediment trap is ready, you can go back to the location with your research vessel. With an acoustic signal, the whole strain is released. The float will come to the surface, and you can pick it up, bring it on board, and then analyze the samples. So as I said, we had sediment trap samples from 5 different locations. And here I'm showing the results of them. So on the x-axis, you will see time in days. So the time is divided into the different seasons, like the northeast monsoon period. There's the spring intermonsoon period, the southwest monsoon, and the fall intermonsoon period. On the y-axis is the flux of lipids in micrograms per square meters per day. And the red lines in these figures, they indicate 114 diol abundances. So what you can see is they are quite abundant at the location of 915. The fluxes are even off screen, so we have to zoom out. And now what you can see is most of the 114 diols, they are produced during the southwest monsoon. And what you also can see is that most of the locations, they have really high abundances during the southwest monsoon, except for the MS4 location, where the abundances are not that high. And this actually fits quite nice because MS4 is just outside of the upwelling area. So we see high abundances of 114 diols within the upwelling area during the southwest monsoon, and not that high abundances outside of the monsoon or outside of the area. And then there's also the blue line, which indicates the abundance of 115 diols. So these are almost the same molecules as the 114 diols, but only the mid-chain alcohol is at a different position. When we zoom in on these lipid fluxes, you can see that they are produced during the southwest monsoon, but they are also produced during the northeast monsoon. And there's not that much difference in the fluxes at the different locations. So it's not like these lipids are specifically produced during the southwest monsoon. And then we can use a ratio of the 114 diols versus the 114 plus C30-115 diols. And we can use this as an index for upwelling. We tested this proxy at the location near the Somalian coast, at the location 905. We had a sediment core which goes back up to 90,000 years ago. And here on the x-axis again is the time, this time in thousands of years. And on the y-axis is the diol index. And you can see that during the Holocene and during the end of marine isotope stage 3 and in the middle of isotope stage 5, you have high values. And at other times, the index is quite low. And when we look at the location nowadays, during the southwest monsoon, you have the low pressure because of the heating of the Tibetan Plateau. But during the glacial periods, snow cover reduced the heating of the Tibetan Plateau, and you did not really get this low pressure formation. And because of that, also you didn't get a strong winds which caused the upwelling. So one way is to compare the diol index with the delta 18O isotope stack from benthic foraminiferals. It's a record published by Lisecki and Ramo in 2005. And this delta 18O of benthic forams, it's a measure for global ice volume. And here the scale is reversed. So the low values of delta 18O, they indicate a large global ice volume. And in this case, it goes together with low diol index values. And at times that there was low global ice volume, like during the Holocene, we see the high index values. So this is a good indication that the diol index is working as a proxy for upwelling in this area. So some other upwelling markers are total organic carbon content, because during the upwelling season you get this high productivity, so you get a high amount of organic carbon. You can look at the content of specific biomarkers like alkanones. You can reconstruct water temperatures, because during upwelling you get these cold deep waters which come to the surface, and you get reduced sea surface temperatures. And you can look at specific foraminifera. So foraminifera, those are protists with calcium carbonate shells, and some of these organisms, they may live during the southwest monsoons. Some of these species might be more abundant during the northeast monsoon. And you can count these organisms in sediments, and you can use a similar proxy like the diol index, where you use you divide the number of southwest monsoon species by the number of southwest monsoon and northeast monsoon species. So we had a number of cultures of Proboscia species producing these long-chain diols, and we noticed that in different species we had different abundances of molecules with double bonds, or the chain lengths varied between the different cultures. So we had 3 different species in culture, and we cultured them at different temperatures. So here in this figure, on the x-axis is the temperature at which the species were cultured, and in this figure on the y-axis is the saturation index. So it is the saturated 114 diol divided by all 114 diols. So as you can see, there seems to be a correlation with temperature here. And the same goes for the chain length index, so the C30 114 diol divided by 28 and C30 diols. So it indicates that with warmer culture temperatures, you get longer chain lengths for the 114 diols. And that's actually pretty cool, because when you can analyze these lipids in ancient sediments, you can calculate the ratio between the C30 and C28 114 diols. And if you know this ratio, you might perhaps be able to reconstruct the temperature at the time that these molecules were formed. So what we did is we analyzed over 200 different surface sediments from different marine locations all over the world, and we analyzed them on the long-chain diol content. So first I will show you the saturated diol index, which I plot here. So on the x-axis is the annual mean sea surface temperature from the location where the samples were taken, and on the y-axis is the diol saturation index from the different samples. And as you can see, for us this was a pretty disappointing result, because there's clearly no correlation between the saturation index and temperature. So if I plot in the results of our cultures, there's really they don't compare. So it looks like the diol saturation index cannot be used as a temperature proxy. And then when we look at the chain length index, I'm plotting the data here. So it may look a bit more optimistic. There's some kind of correlation here between temperature and the chain length index, but it's not great. When I plot in the culture data, well, there's a broad line, but it looks more like the chain length index is more related to the location where the samples are coming from. For example, the Antarctic and the Arctic samples, which are both cold areas, they have completely different long-chain diol index values. So this is also not promising for a temperature proxy. But fortunately, we did not only identify 114 diols, but we also found long-chain diols with alcohol at the C13 position, and long-chain diols with the alcohol at the 115 position. And here I'm plotting the fractional abundances of each of these individual long-chain diols, with the concentrations relative compared to the combined 113 and 115 diol. So now I hope that you agree with me that in this case there is a correlation between the fractional abundances of the 113 diols and the C30 115 diol, but not so much of the C32 115 diol. So this might actually be a more promising temperature proxy. And we propose the long-chain diol index, the LDI, which is an index based on the C30 115 diol divided by the 113 diols plus C30 150 diol concentrations. So the idea is that in cold environments, you will mainly find 113 diols, and the warmer the temperatures, the more of the C30 115 diol you will find. So here I've plotted the LDI values versus the mean annual sea surface temperatures of the locations where the samples were taken. And there is a correlation here between the LDI and the sea surface temperature of with the R square of 0.97, which is really pretty good for a temperature proxy. So this is really good. There are more temperature proxies now. For example, there's a proxy based on long chain alkanones. Here, this is a picture from the white cliffs of Dover. And these white cliffs, they are actually formed by the calcium carbonate shells of corcolithophorid algae. And these algae, they produce what's called alkanones. And we are specifically interested in the ones with the chain length of C37. So these lipids, they have a keto group at the C2 position, and they may differ in the number of double bonds in their carbon chains. So it was Brussels et al. In 1986 who analyzed the alkanones together with 18O data from forums, and it was known that these delta 18O, it is related to temperature. So they analyzed the sediment core, and they noticed that the double bonds in the alkanones, it seemed to be related to the delta 18O. So that might actually mean that the alkanones also could be used as a proxy for sea surface temperature. Later, Prahl and Wakeham, they slightly modified the proxy that was proposed by Brussels et al. And they divided the C37 with 2 double bonds by C37-2 and C37-3. And it also has a very nice correlation with annual mean sea surface temperatures. This is in sediments, but that's also the case in samples taken from the surface waters. So this is filtered seawater where they analyzed the UK37. And there's a really nice correlation. This is a figure by Rosa-Mallet and Prahl in 2013, where they are showing sediment trap data of long chain alkanone fluxes. So the triangles, they show the fluxes of alkanones during specific months. And what you can see in this figure is that at some locations, long chain dials are formed all year long, but at other locations, they might be produced only during specific months. And at locations close to each other, the months of long chain dial production might be completely different. So actually, it is quite surprising that the UK37 is giving such a strong correlation with mean annual sea surface temperature. And besides long chain diols and alkanones, there's another group of lipids which are used for sea surface temperature proxies, which are the glycerol dialkyl glycerol tetraethers, or abbreviated as GDGTs. These lipids are produced by Archaea, and Schouten et al. They noticed in 2002 that these lipids might be a good proxy for temperature, and they introduced the TEX86, which is based on the number of cyclopentane rings in these molecules, and including an isomer of Crenarchyl, which has this cyclohexane ring. So the TEX86 also gives a good correlation with sea surface temperatures. And there was a study by Lopez dos Santos et al. In 2013, where they analyzed both the UK37, the TEX86, and the LDI on one sediment core off the coast of Australia. And here you see the results. On the x-axis, it's the age of the sediment samples in kilo years. So on the y-axis is the reconstructed temperature of the different proxies. And you may notice that there is quite a difference between the reconstructed temperatures of the different proxies here. in the study by Lopez dos Santos et al., they also looked at the temperatures reconstructed by a modern analog technique of foraminifera assemblages. And this, again, is based on foraminifera, and it enables them to reconstruct temperatures of different periods. So they had the temperature of the highest, the warmest month of these samples. They reconstructed temperatures of the lowest month during a year, and they reconstructed mean annual sea surface temperatures. And when they compared it with the different proxies, it looks like the LBI is correlating really well with the maximum temperature. The TEX 86 is correlating really well with the minimum temperatures. And the UK 37 is giving a good correlation with the reconstructed mean sea surface temperatures, annual mean sea surface temperatures. So it looks like the three proxies are giving temperatures of different months or different seasons. And in that way, the different proxies, they give complementary data, which is really useful. So in this sediment core off the coast of Australia, they also identified C32-115 diols. And while in the marine samples that we analyzed, the fractional abundance of C32-115 was often around 0.2, 0.1. In the sediment core, the fractional abundances of these long chain diols were sometimes much, much higher, to 0.5 or even higher. And this is quite unusual for the marine environment. But in a study where we looked at lake sediments, we did find such high relative abundances of C32-115 diols. So it looks like there is a freshwater signal in the sediment core. So nowadays, the location of the sediment core is quite far off the coast, but during the glacial periods, the sea level was much lower, and actually the shelf of Australia was dry at that period, and there was a river going almost all the way to the location of the sediment core during the glacial periods. So what we are seeing here in the C32-115 record is periods where the sea level was low enough that the shelf was actually dry. And we can compare this with the bit index, which is an index based on branched versus isoprenoid tetraethers. So again, these are GDGTs, where the branched GDGTs have mainly a terrestrial source, and the isoprenoid GDGTs, which is in this case is granarcheal, it has a marine source. And the bit index can therefore be used as a proxy for terrestrial input, in this case in the marine core. And since the two records are really, they look the same, it looks like the C32-115 diol fraction is a promising proxy for freshwater input. So although I'm not that familiar with isotope analysis myself, it does have an important use in paleoenvironmental studies, so I do want to cover it in this talk. So when you are looking at the molecule, we might have atoms with different isotopes. For example, carbon comes as 12C. There's like 1% is in the form of 13C, and there's a fractional abundance of the radioactive 14C. You can use carbon isotopes, for example, in incubation studies. So what I did is I had a project where I looked at deep sea waters, and we brought deep sea water on deck and added labeled material to it to see what the deep sea waters were lacking. So we added bicarbonate to see if we could create productivity in the deep sea waters by adding carbonate. We added glucose, where we added both energy and carbon, and we added amino acids, where we added both energy, carbon, and a nitrogen source. And in addition to that, we had the control. So we added this labeled material to bottles with deep sea waters. We let them stand for a day, and then we filtered the material and analyzed the 13C. So what we noticed is that when we added the bicarbonate, so only the carbon source, we did not really see that 13C was incorporated in any of the biomarkers that we analyzed. But when we added labeled glucose or amino acids, we saw a strong 13C signal in fatty acids and even in some sterols, which indicates that when you add energy and also when you add nitrogen, you can increase the growth of the organisms in the deep sea. So it might be that energy and nitrogen is lacking in the deep sea. So biosynthesis actually prefers 12 carbon above 13 carbon. So when you have biosynthesis, you will see that relatively you get more 12C incorporated. And this is called the isotope fractionation factor. So the ratio of heavy versus light carbon in your biomass divided by the ratio in heavy versus light isotopes in, for example, carbon dioxide is called the isotope fractionation factor, alpha. Here I'm showing the carbon delta 13C values. This is the abundance of 13C in grasses, and it is indicated as a delta 13C, which is the fractional abundance of 13C relative to a standard. And here I'm showing grasses which are produced by, it's from a study by Serling et al. In already 1997. And grasses which use the C3 photosynthetic pathway, they are relatively depleted in 13C, so they have lower 13C values. And the plants that are using this C3 pathway are almost all of the trees, most of the shrubs, herbs, and forbs, and grasses which grow in cool seasons. And the C4 grasses, they are mainly warm season grasses. So Serling et al. Analyzed the isotopic fraction of grasses, and they also analyzed teeth from mammals. And there is a fractionation effect, so the 13C values in the teeth is different from the grasses that these mammals had been eating, but the difference is similar for the mammals that were eating C3 dominated grasses and for the mammals that were eating C4 dominated grasses. So by analyzing the delta 13 of teeth, you can reconstruct the diet of these mammals. So when they analyzed the 13C content of teeth from 8 million years ago, they found 13C values which are similar to the ones of the mammals that only had the C3 dominated diet. So based on these results, they concluded that at the time 9 million years ago, there were mainly C3 grasses. So they analyzed teeth over a range of time, so from almost 20 million years ago to now. And they noticed that around 6 million years ago, the delta 13C in these teeth suddenly changed. So it looked like the diet of these mammals changed from C3 to more C4 grasses. And they concluded from this that probably, well, the vegetation had changed at 6 million years ago, and they thought that it might have been related to a change in the carbon dioxide concentrations, which became lower around 6 million years ago. And C4 grasses, they are better capable of living under low CO2 concentrations. There are more things you can do with carbon isotopes. So here I'm showing a map of Africa, and the color is indicating C4 versus C3 plants. So green is an indicator of C4 plants, and blue is an indicator of C3 plants. And Shanahan et al., they did a study on a sediment core from a lake, and there they analyzed the carbon isotope composition of molecules. These are alkanes, which are leaf wax lipids. So they analyzed alkanes in the sediment core, and the 13C of these alkanes. And what they noticed is that around 15,000 years ago, on this location, they noticed a change. And it seemed like at that time, the vegetation changed from grassland to more forest vegetation. So that might have been a climate change which caused this change. So in addition to carbon isotopes, you can also analyze hydrogen isotopes or deuterium isotopes. Actually, that's quite complex, and I'm going to give an easy rationale behind using these isotopes. So in general, deuterium, so that's the heavy isotope of hydrogen. Deuterium prefers to stay in solution. So during evaporation, the hydrogen is mainly the H1 and not much of deuterium evaporates. So the clouds are relatively light, so they have little deuterium concentrations. And when it starts raining, most of the deuterium will drop down as soon as possible. So in time, the clouds, they will get relatively light in their isotope composition. So that means at the time that the clouds are formed at high temperatures, the isotopes, the deuterium isotopes, is relatively high. And the more you get into land, the lighter the isotope value is of the clouds and then also of the rain. So Shanahan, in their study, they also analyzed the hydrogen isotopes of the leaf waxes, so that's delta D values, and they noticed quite heavy samples, so high delta D values in the oldest sediments. And then again, at around 15,000 years ago, they saw a change up to samples that were more depleted in delta D value, so the hydrogen became lighter, which they related to more humid periods. But then at around 5,000 years ago, the delta D values, they decreased again. So this is a slightly different picture than what they showed from the delta 13C values, where the change to C3 plants, they remained after 15,000 years. They did not decrease like we see with the delta D values. And when they looked at the CO2 concentrations, they concluded that probably the higher concentrations of CO2 after 15,000 years ago, they may explain why, although it became drier in the last 5,000 years, why the vegetation stayed more to the C3 plants. Another thing you can do with hydrogen isotopes is a study by Schouten et al. In 2006. They analyzed alkanones, which were grown in under conditions with different delta D values. So the deuterium concentration in the water, it changed for the different samples that they analyzed. And they noticed that there was a correlation between the delta D in the alkanones, in the lipids, and it correlated with the delta D in the culture media. So it looked like the species producing the alkanones, they took up the delta D from the water. They also noticed that there was a correlation between salinity and the delta D values in the alkanones. So cultures that were grown under higher salinity, they had slightly less depleted or heavier delta D values in their alkanones. And in itself, it did not really came as a surprise, because the way they made the different culture media was that for the more saline culture media, they evaporated the culture media, which may have removed the lighter hydrogen isotopes from the media. And in order to get lower salinity culture media, they added culture pure water, which also had a lower delta D value. However, when they calculated the fractionation factor for the different samples, which is quite complex and I'm not going into detail about that, but they noticed that for the different samples, the fractionation factor was different. So the different concentrations of delta D could not explain the delta D in the alkanones. So there was something else which caused a difference in the fractionation, and they checked the temperature, which did not really have a alkanone fractionation effect. They checked salinity, and they noticed that the fractionation effect of alkanones was positive with salinity. So with higher salinity, indeed, the fractionation effect was higher. And they compared it with the growth rate of the cultures. And with the higher growth rate, the fractionation effect was lower. So based on these results, they suggested that the delta D of alkanones, because there's a correlation with salinity, might be useful as a proxy for salinity. Now, this worked pretty good in the culture experiments, but Weiss et al. In 2019, they analyzed several surface sediments and analyzed delta D, the hydrogen isotopes in alkanones again, and they compared it with annual mean salinity, and they also analyzed delta D of the water, of the surface waters of these locations. And although they did see a correlation between the delta D in the alkanones and the salinity, so there was a positive correlation, when they calculated the fractionation effects of these samples, well, there was not really correlate. The fractionation index did not really change for the different samples. So what they suggested was that maybe the species, different species have different fractionation effects, and this might have hampered to make a global correlation line. However, several studies have shown actually that the delta D in alkanones might be useful as a proxy for salinity. So although there are challenges, the hydrogen isotopes in alkanones might actually still be a good proxy for salinity. This is a study by Lato et al. It is the northern Gulf of Mexico, where you also have a small salinity gradient. When further off the coast, you will get a higher salinity. And they analyzed the delta D in long chain diols and in alkanones. And indeed, they saw a slight correlation between the delta D in the alkanones and salinity. They saw a slight correlation between the delta D in the C3115 diol and salinity. But the main and the most intriguing part was that the C32115 diol, they had much lower delta D values, and there was not much of a correlation. But actually, that makes sense, because if you assume that the C32115 diols, if they are produced in freshwater conditions, this would result in low delta D values, and you would not see a correlation in a marine environment, because that's not where they are produced. Well, there are plenty of other things that I can talk about. I can go on for hours, but I think it's time to stop now. And one thing, what really matters when you are having all these climate proxies is, well, you can reconstruct everything you want, but the most important thing to give the whole picture. So what's happening when the climate is changing? So don't just show that there is a change in maybe 1 degree or a bit of salinity, but show that changes in the climate might have far-fetching consequences for life on Earth. So that's the main message that you should get from climate reconstruction experiments. And that's the end of my talk for now.
Okay, Sebastian. Thank you very much for this wonderful talk, coupling several tools, as traditional ones with innovative biomarkers, also phylogenetic trees, isotopes, all together to elucidate the environmental conditions and also age. So we were talking about temperature, salinity, and so on. Thank you very much. So I will pass direct to some questions we have here. The first one is from Luisa from UF University. And she asked, did you notice any influence of the lithology related to the diols index, or any influence related to the age of the rocks?
I'm sorry. Can you repeat the question?
If the diol index, if it suffers some influence of the lithology, the characteristics of the rocks from the continental. So if there is any influence related, or the lithology of the rocks, of the age of the rocks.
We do notice that there might be a slight effect of degradation on the long-chain diol index. So it might be that the index is affected by degradation. So that gives an error in the reconstructed temperatures, but it's a relatively small error.
Okay, perhaps the lateral, the runoff influence because of diatoms from freshwater, but not from an influence from lithology, no?
No, not really.
Okay, the second one from Giovanna, UFAM University, here from Brazil. Thank you, Sebastiaan Rampen, for the presentation. Have sulfurized C27 esterols been used as indicator of euxinic environmental, natural or eutrophic conditions?
I must admit, I don't know about it. I'm sorry, I cannot answer that question.
About C27 esterols. Okay, that's not this biomarker you worked with. Okay, third question from Professor Débora Azevedo from University Federal of Rio de Janeiro, our president from ALAGO. Did you detect these long chain diols with GCMS or which equipment?
Yeah, we use a GCMS, and what we do is we use selective isotope measurements, because sometimes the concentrations are really low. So we make mass chromatograms of specific ion fragments, and then you can get a really good signal. And that's also the way how you can distinguish between the 113, the 114, and the 115 dials, because with the same chain length, they elute at the same time, but by looking at different mass fragments, you can distinguish between them.
Okay, and I complement her question. The column is 30 meters column is enough, sufficient, or a much
That's
longer
what we
column?
normally use. We normally use
Okay.
like a CPCL5 column or something similar to that, and 25, 30 meters should be enough.
Okay, thank you. One more question from Professor Renato Carreira from the University PUC from Rio de Janeiro. Dear Sebastian, many thanks for the talk. The question is, what are your opinion regarding the best biomarker for pyrogenic carbon from the last 2 million years? Is the black carbon, the PAH, or the BPCA, or another one?
Oh, that's another question that I'm not really familiar with. No, I'm sorry. It's not something I can answer.
Yeah, we would like to have always the perfect biomarker, no, Sebastian? And actually, my question also for you, watching your results, I was wondering how to figure out the better way to work with the positive and negative points for each biomarker and try to choose the best one. Actually, because they are expensive analysis. And regarding your results from diatoms, I have one question. What is the better approach to do macroscopic analysis for diatom from the fistulas in the sediments, or doing the biomarker analysis? What do you prefer to choose?
For me, I'm a real biomarker, and that's what I do. So I like to break everything up, crush everything, and just look at the biomarkers, because it is quick and you can look lots of things in a relatively short time.
Yeah, and we have to think about the preservation rate, which kind of the refractory material in the macroscopic analysis, or the organic, the molecule, one more label will be more preserved in the sediments. It's a huge question. And also, I thought very interesting when you compare the temperature proxies using Alkanone, the UCA 37, the GDGT, the TEX 86, and you include a comparison with your index, the long chain diol index. And I watching some papers that they are not always telling us the same behavior, and we have to interpret what happens at that period, because when you use core studies, we are studying different periods of the environmental conditions, and we expect that the sea level oscillation, the water mass oscillation, actually because of temperature, different temperature mass water, and also the upwelling occurrence. So we have different conditions that print the signature for those molecules, and it's really hard to do the interpretation. And you show us that sometimes it is not the annual temperature, but some month of the year will print the signature specific for each organisms. If there's a diatoms, if there's a foraminifera or an algae. And that is very interesting. That's really very interesting. We can not close, but we can choose another kind of interpretation because we have this lateral influence, so much influence, physical, chemical, and environmental conditions, and we don't we really always do a right interpretation. And so what I conclude, it's not a question, is that we do need to do different markers. It's expensive, but I think, do you agree with me? It's a really good deal to do different markers.
Oh yeah, definitely. But there's also the issue that, for example, the GDDTs, they do not always indicate the same period or even the same water level. Sometimes the GDDTs, they reconstruct surface water temperatures, but sometimes they can be used to reconstruct the deeper water layers, because the organisms, they sometimes live in different water masses, or they live during different periods, which is also a challenge. So it's not always clear which period of the year you are reconstructing, and it's also not always clear if this is constant or if it might change during different conditions.
Yeah, that's a nice argument to our supporters to give us more money to do much more analysis.
Yeah.
And you show at the end that the isotope composition, not only carbon, but the deuterium and the oxygen, help us a lot to understand environmental conditions. That's really, really, really nice.
Yeah, it's always good to use different proxies at the same time, because one proxy might not show you the complete picture, and it may even give you the wrong picture if you don't see everything at all. Like I showed for the 13C, you might think that it indicates aridity, but when you use the delta D values as well, you may get a different picture, and it might not only be aridity, but also the CO2 concentration, which plays a role.
Yeah, those results are really interesting and explain why not you have the same results like the literature tell us. Okay, we have two more questions. You have the time running. One question from Alexandre, a research from Petrobras company. Did you ever consider to incorporate sulfur isotope to your work? Which kind of information would you expect to obtain if you consider to use the sulfur isotope?
I have no doubt that it is very interesting, but I'm mainly looking at living organisms first, and then see how they react in sediments. But this would be kind of a different way of looking at biomarkers, which is not really something that I'm experienced in. So I don't know. Okay,
should be very interesting, yes. And the last question, and please, Sebastiaan, from Jefferson from University Federal of Goiás. Can you comment about the process of establishing a ratio as a temperature proxy? What were the initial facts of evidence that led to the choice of a specific biomarker?
So there are different ways to come to that. So you always need different approaches. So in case of the LDI, well, first we thought we had a good temperature proxy with 114 diols because it worked in cultures. But then when you go to the real environment, it seems like it's not working. But in case of the LDI, there seems to be a good correlation with temperature. But in that case, it would be nice to have the source organisms in culture, so you can see whether it is only temperature which affects the proxy, or maybe also other conditions like salinity or nutrients might also affect the proxy. So there's not one way to establish whether proxy is working or not, and it is always good to look from different angles at a proxy.
Okay. So we have run out of time. Okay. I thank everyone for participating. If you still have some question or it has not been answered, we can see the possibility to asking it by email. But Sebastiaan, it was really a pleasure to have you here, giving us the opportunity to share your knowledge in those different kinds of biomarker, isotopic, and also phylogenetic analysis. Okay, thank you very much. I just have one last announcement, last message. Please don't miss next webinar, ALAGO webinar, on May 27, next Thursday at 6 p.m. with the lecture Digital Transformation in Geoscience, Challenges, Technologies, and Opportunities. The speaker will be Gustavo Robichês from PUC Rio de Janeiro. And also follow us for the next ALAGO event in June about analytical methodology in organic geochemistry. Okay? So thank you, everyone, and good evening for all. Thank you, Sebastiaan.
Thank you for the opportunity, and goodbye.
Okay.